All right, when you're in your FRCS Viva, you've got to be prepared to discuss the landmark papers that have really shaped our practice. Today, we are tackling one of the absolute biggest, the 2018 Katsanos meta-analysis on paclitaxel devices. This is the paper that uncovered a really critical long-term mortality signal, and you can bet the examiners will expect you to dissect its findings with absolute precision. So let's get into it and structure that perfect consultant-level answer. Right, let's start. Look, a core skill at this level is being able to critically appraise these huge papers. What the examiners are really testing is your ability to clearly explain the clinical question, the methods, the key results, and most importantly, the impact on your practice. We're going to model that exact structure right now for the Katsanos paper, so you can deliver it clearly and confidently, even under pressure. First things first, what was the clinical context? You know this better than anyone. Femoropopliteal restenosis. It remains the biggest headache after we do any endovascular intervention. This whole area of research was really driven by one thing: finding a lasting solution that could do better than just standard angioplasty and bare metal stents.
Yeah, standard angioplasty and stenting were, and let's be honest, still are, limited by neointimal hyperplasia. That's what leads to those high rates of having to go back in and retreat the same spot. So along came Paclitaxel-coated devices, or PCDs. The whole idea was to fight this problem by delivering an antiproliferative drug right to the vessel wall. And the early data on reducing restenosis, it looked fantastic.
So what's the magic ingredient? It's paclitaxel. It's a cytotoxic drug that works by messing with microtubule function, which basically stops the cell cycle in its tracks. Its anti-restenotic properties are powerful, which made it seem like the perfect choice to stop that smooth muscle cell growth that causes all our other treatments to fail. But here's the thing, great efficacy doesn't mean a thing without proven long-term safety. And that brings us to this meta-analysis. It was designed for one specific reason, to look at that long-term safety signal across all the available trial data. This is the crucial point you have to make to the examiner. The real power of this study came from its sheer scale. The authors didn't just look at one or two trials, they pooled patient data from 28 separate randomized controlled trials. You see, this created the biggest, most solid dataset available to really dig into this question of long-term safety. And that involved a massive number of patients, 4,663 to be exact. With a sample size like that, whatever they found was going to have serious statistical weight. It was going to be impossible for the clinical community and the regulators to just ignore it. So what were they looking for? This is key. The primary safety endpoint was all-cause mortality. For a safety analysis, that's the gold standard. It's robust, it's unbiased, because you don't have to argue about what caused the death. The question was incredibly simple. Did more patients die in one group than the other? Period. Okay, let's get to the results. The analysis started where most of the individual trials had reported their own main outcomes, which was at the 1-year mark. This gives us the baseline for the whole safety story. And at 1 year, everything looked great. It was totally consistent with what the individual trials had shown. There was no safety signal at all. The crude risk of death was identical, 2.3% in the paclitaxel group and 2.3% in the control group. So at this point, it seemed like these devices were both effective and safe. But, and this is a huge but, the real value of a meta-analysis like this is its ability to pool that longer-term follow-up data, the data that you just couldn't see in most of the individual studies. And this is where the story takes a sharp turn, and that critical safety signal pops up. By year 2, a statistically significant mortality signal appeared. All of a sudden, the death rate was 7.2% in the paclitaxel group versus just 3.8% in the control group. That's a 68% relative increase in the risk of death. Suddenly, that reassuring safety profile was in serious doubt. And if you translate that into a number needed to harm, it was 29 at 2 years. That's a clinically significant number. It suggests that for every 29 patients we treated with a paclitaxel device, there was 1 extra death. And it didn't stop there. The signal just got stronger the longer they looked. At 5 years, the mortality rate in the paclitaxel group hit 14.7%. Compare that to 8.1% in the control group. We're now talking about a 93% relative increase in the risk of death. And that brought the number needed to harm all the way down to just 14 at 5 years. So the big takeaway here is that this wasn't some static risk. The data suggested a persistent and actually worsening mortality signal over time. So when you see a finding like this, the next logical question, the one the examiner will definitely ask, is why? What's the mechanism here? How could a device that we apply locally have a systemic, long-term, deadly effect? Now, because this was a meta-analysis, a review of trials that were already done, it couldn't prove what was causing this. But the authors did propose several pretty plausible ideas based on the drug's pharmacology and how these devices work. So the main theories they floated were, 1, some kind of late toxicity from the drug itself, which can hang around in the tissue for a long time. 2, systemic embolization, basically tiny microparticles of the drug breaking off and causing inflammatory effects downstream. And 3, a potential dose-dependent relationship, because their analysis hinted at a link between higher drug doses and higher mortality.
So, how do you pull all of this together into a sharp, structured answer for your exam? You've got to summarize the findings, acknowledge the limitations, and then state the impact on our clinical practice.
Your starting point should be the paper's own conclusion. They didn't mince words. It was unambiguous. There is an increased risk of death, and further investigation is urgently needed. That clear, definitive statement is what set off alarm bells and triggered immediate action from regulatory bodies like the FDA. Okay, so if you're asked in the exam, here's the safe, consultant-level answer. It's this. The 2018 Katsanos meta-analysis of 28 RCTs was designed to evaluate the long-term safety of Paclitaxel-coated devices. Its key finding was a statistically significant jump in all-cause mortality starting after 1 year. The numbers you need to quote are the crude death rates, 7.2% versus 3.8% at 2 years, and 14.7% versus 8.1% at 5 years. This gave a 5-year number needed to harm of just 14. Now, while the exact mechanism isn't known, the leading theories are things like late drug toxicity or systemic embolization. The paper's firm conclusion that a clear mortality signal exists had a profound impact on practice and led to urgent regulatory reviews and a demand for more trial data.
At the end of the day, this paper is just a powerful case study. It really gets at that tension between pioneering new, innovative treatments and the slow, methodical work of post-market surveillance. It highlights our professional duty to demand and to critically analyze long-term safety data, even for devices that look great in the short term. Because in medicine, the final verdict is very rarely delivered early.